Welcome back to the deep dive. Today we're tackling the final step in the central dogma of genetics.
That's right.
We've seen how DNA holds the code, how transcription creates that messenger RNA, the mRNA.
Yep.
So now, the big question, how does that mRNA sequence actually get turned into, well, into proteins, into the stuff that does things?
Exactly. That's our mission today. Decoding that mRNA blueprint, we're diving into the molecular machines and the process itself, how the cell reads this ribonucleotide sequence to build a polypeptide chain.
And that chain then folds up
Right.
into a complex 3D shape.
Precisely. That specific three-dimensional structure is what makes a protein functional. It's really the basis for, well, almost all biological activity and diversity.
Okay, so this is the shortcut, folks. If you want to really get the machinery, the process, and even a bit of the history behind turning genetic code into functional proteins, stick around. Let's get into it. First off, we need something to bridge the gap between nucleic acids, the language of mRNA, and amino acids, the language of proteins. Francis Crick guessed this back in '57, didn't he? He called it an adapter.
He did, yeah. Brilliant intuition. And that adapter turned out to be transfer RNA, or tRNA.
tRNA.
It's quite elegant, really. It has two crucial parts. One end has the anticodon 3 bases that pair up with a specific codon on the mRNA.
Okay.
And the other end, it carries the exact amino acid that the mRNA codon is calling for. It's cognate amino acid, so it's literally translating the code.
The molecular dictionary. Got it. And this all happens on the ribosome, the workbench, as you called it.
That's the place, the ribosome. It's this huge, complex structure made of a large and a small subunit. They differ slightly between bacteria and eukaryotes, you know, the 70S versus 80S thing.
Right, those Fedberg units that don't just add up.
Exactly. But the truly mind-blowing part, the thing that really changed our view, wasn't just the structure, but what does the work inside?
We always assumed proteins did all the catalytic work in the cell, right? Enzymes.
Pretty much. But the Nobel-winning work on ribosome structure showed something amazing. It's the ribosomal RNA, the rRNA, that actually catalyzes the most critical step, forming the peptide bond between amino acids.
So the ribosome is an RNA enzyme.
A ribozyme.
A ribozyme, exactly. The proteins are there, mostly on the surface, kind of scaffolding and helping things along, fine-tuning. But the core catalytic engine is RNA. Huge implications, especially for thinking about the origin of life.
Wow. Okay, but before translation can even start, those tRNAs need to be loaded with the correct amino acid. That sounds like a critical checkpoint. Get that wrong, and everything downstream is messed up.
Absolutely crucial. It's called charging, or amino acylation. And it's done by these incredibly specific enzymes, the amino-acyl tRNA synthetases.
How many are there? 20. One for each type of amino acid. Their sole job is to recognize one specific amino acid and attach it, chemically link it, to only its corresponding tRNA molecules. The fidelity here is paramount.
And it's a strong link.
Oh yeah. It's a two-step process that uses ATP for energy. The result is a high-energy ester bond connecting the amino acid to the tRNA's 3' end.
And that energy is used later.
Exactly. That bond holds the energy that will be used to actually form the peptide bond during translation. So the accuracy of these synthetases is just fundamental to the whole process.
Okay, let's walk through the process itself. Maybe start with bacteria since it's a bit simpler.
Good idea. So inside the ribosome, there are 3 key sites, sort of like docking stations for the tRNAs. The A site for aminoacyl, where the next charged tRNA comes in. The P site for peptidyl, which holds the tRNA carrying the growing polypeptide chain. And the E site for exit, where the now uncharged tRNA leaves.
APE sites. Got it. How does it start? Initiation.
Initiation needs a few things. The small ribosomal subunit, the mRNA template, a special initiator tRNA, and some protein helpers called initiation factors, plus energy from GTP.
And how does the ribosome find the exact starting point on the mRNA in bacteria?
Ah, that's the Shine-Dalgarno sequence. It's a specific sequence, AGAG, upstream of the actual AUG start codon on the bacterial mRNA.
And it pairs with the ribosome.
It base pairs directly with a complementary sequence on the 16S rRNA within the small subunit. It basically anchors the ribosome in the perfect spot, ensuring the AUG start codon is positioned correctly in the P site. So that's the reading frame.
So the initiator tRNA carrying N-formyl methionine in bacteria goes straight to the P site.
Directly to the P site. That's unique to initiation. Then the large subunit joins, the initiation factors leave, and you have a complete ribosome ready for the next phase. Elongation.
Elongation. Adding amino acids one by one. How does that cycle work?
It's a repeating 3-step dance. Step 1. A charged tRNA, matching the codon exposed in the A site, is escorted in by an elongation factor protein called EF2 using GTP for energy.
Okay, tRNA in the A site.
Step 2. Peptide bond formation. This is the ribozyme action. The rRNA catalyzes the transfer of the polypeptide chain from the tRNA in the P site onto the amino acid of the tRNA in the A site. The chain just grew by 1.
And the energy for that bond came from that initial charging step.
Right, precisely, from that high-energy ester bond. Step 3. Translocation. The whole ribosome shifts exactly 3 nucleotides along the mRNA. This needs another elongation factor, EFG, and more GTP.
So what happens to the tRNAs during that shift?
The now empty tRNA that was in the P site moves to the E site and exits. The tRNA that was in the A site, now carrying the longer peptide chain, moves into the P site, and the A site is now empty, exposing the next codon.
And the cycle just repeats.
Yeah.
A site filling, peptide bond forming, translocation.
Over and over.
Over and over. And fast. In bacteria, maybe 15 amino acids per second.
Until it hits a stop signal.
Right. When a stop codon UAG, UAA, or UGA enters A site, there's no tRNA that recognizes it. Instead, proteins called release factors bind.
Release factors. RF1 or RF2, depending on the specific stop codon. Their binding triggers the ribosome to cut the polypeptide chain free from the tRNA in the P site.
And the protein is released?
Finished protein floats away. Then another factor, RF3, helps the whole complex ribosome subunits mRNA, remaining tRNA, to disassemble, ready to start again somewhere else.
And cells make this even more efficient with polysomes.
Polysomes, yeah. Or polyribosomes. It just means multiple ribosomes translating the same mRNA molecule simultaneously, like an assembly line. As soon as one ribosome moves down the mRNA a bit, another one can hop on at the start. Very efficient.
Okay, let's switch to eukaryotes. Similar core process, but some key differences, right? Especially around initiation.
Definitely. Big difference is location transcription in the nucleus, translation out in the cytoplasm. And eukaryotic ribosomes are slightly larger, ADS, but initiation is quite different. No Shine-Dalgarno sequence.
So how do they find the start?
It's usually cap-dependent. The small ribosomal subunit, along with a bunch of eukaryotic initiation factors, or EIFs, first recognizes and binds to the 5' cap structure on the eukaryotic mRNA.
The M7G cap.
That's the one. Then this complex typically scans along the mRNA, moving downstream until it finds the first AUG codon.
Scanning. Okay.
Yeah.
Is there anything that helps it find the right AUG?
Often, yes. A sequence around the AUG called the Kozak sequence, AGNNAUG, helps increase the efficiency of initiation at that site. And the initiator tRNA in eukaryotes carries regular methionine, not formal methionine.
And you mentioned this idea of closed-loop translation. Sounds intriguing.
It is pretty neat. Proteins that bind to the poly-A tail at the 3' end of the mRNA can actually interact physically with the cap-binding proteins at the 5' end.
So the mRNA forms a circle.
Effectively, yeah. A closed loop. The thinking is this has a couple of advantages. One, it's a quality control check, ensures the mRNA is intact, has both a cap and a tail, before the cell invests energy translating it. Two, it could make reinitiation more efficient. A ribosome finishing translation at the 3' end is already right near the 5 cap, ready to start another round.
That makes a lot of sense. And all these details, especially the ribozyme part, have become much clearer with new technology, right? Like cryo-EM.
Oh, absolutely. The high-resolution structures from researchers like Nola, Ramakrishnan, and others have been revolutionary. Seeing the ribosome at near-atomic detail confirmed RNA's central catalytic role.
What else did those studies show?
They revealed just how dynamic the ribosome is. It's not a static machine. It undergoes large conformational changes, especially during translocation. And interestingly, it seems to harness random thermal energy Brownian motion to drive some of these movements, with the elongation factors kind of rectifying that motion, making it directional. Energy is used, but maybe not quite how we originally pictured it for every single movement.
Amazing detail. Now, to really appreciate why this whole protein synthesis machinery is so fundamental, maybe we should touch on the history. How did we figure out genes make proteins in the first place?
Yeah, that's a great story. It really started clinically with Sir Archibald Garrod back in the early 1900s. He studied inborn errors of metabolism.
Like alkaptonuria.
Exactly, AKU. People with AKU accumulate homogentic acid because a specific step in a metabolic pathway is blocked. Garrod connected this inherited condition to a faulty chemical reaction, suggesting genes control enzymes. Phenylketonuria, PKU, is another classic example he studied.
So the first hint that genes relate to biochemical functions. But the real proof came later.
Much later, the 1940s. Beadle and Tatum's work with Neurospora, the bread mold, that was definitive.
What did they do?
They deliberately created mutations using X-rays, then looked for strains that could no longer grow on basic minimal medium. They found mutants that needed specific extra nutrients, like a particular amino acid, say arginine.
And they figured out why they needed it.
Yes, by testing different precursors in the arginine synthesis pathway, they showed that a specific mutation blocked one specific enzymatic step. If you supplied the chemical after the block, the mold could grow. This led them to the powerful one gene, one enzyme hypothesis. A gene specifies an enzyme.
A landmark idea. But it needed a slight tweak, didn't it? Based on sickle-cell anemia?
It did. First, Linus Pauling showed hemoglobin from sickle-cell patients, HbS, behaved differently electrically than normal hemoglobin, Hba. It had a different charge.
Okay, so the protein itself was different.
Right. But the clincher was Vernon Ingram's work a few years later. He used a technique called peptide fingerprinting to pinpoint the exact difference.
And what was it?
Just one single amino acid change. At position 6 in the beta-globin polypeptide chain, HbS has a valine instead of the normal glutamic acid.
One single change caused by one gene mutation altering the protein's properties.
Exactly. And since many proteins, like hemoglobin itself, are made of multiple different polypeptide chains, 2-alpha, 2-beta, and Hb, each coded by its own gene, the hypothesis was refined to one gene, one polypeptide chain. That's pretty much where we stand today.
Okay, so the gene dictates the polypeptide chain. Now let's talk about the structure that chain takes on. You mentioned folding.
Right. The linear chain, the polypeptide, isn't usually functional itself. It has to fold into a specific 3D shape. We talk about 4 levels of protein structure.
Primary structure first.
Primary is just the sequence, the linear order of amino acids directly determined by the mRNA sequence, which came from the gene.
Simple enough? Then secondary.
Secondary structure refers to local repeating folding patterns. The 2 main ones are the alpha helix, like a right-handed coil, and the beta-pleated sheet, where strands lie side by side. Both are stabilized by hydrogen bonds between the backbone atoms, not the side chains.
Okay, helices and sheets. Then tertiary structure.
Tertiary is the overall, final, 3-dimensional shape of a single polypeptide chain. This is the money level, really, because the tertiary structure determines the protein's function.
What holds that shape together?
A whole range of interactions. Hydrophobic interactions are key non-polar side chains, burying themselves inside, away from water. Hydrophilic polar side chains tend to be on the surface. Also, hydrogen bonds between side chains, ionic bonds, and sometimes strong covalent disulfide bonds between cysteine residues.
And quaternary.
Quaternary structure only applies if the final protein is made up of more than one polypeptide chain, like hemoglobin. It describes how these multiple subunits, or protomers, fit together to form the complete functional complex.
And this folding process, it can start even while the protein is being made.
It often does, yeah, co-translational folding. But it's not always spontaneous or perfect. That's where chaperones come in.
Molecular chaperones, what do?
They're helper proteins. They bind to unfolding or partially folded polypeptides and prevent them from aggregating incorrectly. They help guide the protein along the correct folding pathway to reach its final functional state. They don't dictate the final structure, but they help it get there efficiently and correctly.
And if folding goes wrong anyway, if a protein gets terminally misfolded?
Eukaryotic cells have a robust quality control system for that. Misfolded proteins get tagged with a small protein called ubiquitin.
Ubiquitin tagging.
Right. That ubiquitin tag is like a label that says, "Destroy me." It targets the misfolded protein to a large molecular machine called the proteasome.
And the proteasome.
Is basically a protein shredder. It unfolds the tagged protein and chops it up into small peptides, recycling the amino acids, keeps the cell clear of potentially toxic junk protein.
One last structural concept, protein domains. What are those?
Domains are distinct structural and functional units within a larger polypeptide chain. Think of them as modules, maybe 50 to 300 amino acids long, that often fold up independently.
And they have specific jobs.
Often, yes. One domain might bind DNA, another might have catalytic activity, another might bind a signaling molecule. A single protein can be made of several different domains, each contributing to its overall function.
And this relates to evolution, exon shuffling.
Exactly. Walter Gilbert proposed this idea. Since exons in genes often correspond quite nicely to these functional protein domains, He suggested that evolution can create new proteins relatively quickly by mixing and matching existing exons through processes like recombination.
So you shuffle the exons, you shuffle the domains, and potentially get a new protein with a novel combination of functions.
Precisely. The LDL receptor gene is often cited as a great example of this. It
Mhm.
looks like a mosaic of domains borrowed from other proteins. It's a powerful way to generate novelty.
So to wrap up, we've followed the genetic message from mRNA right through the ribosome, seen how that polypeptide chain gets built step by step and how it folds into these intricate structures. We've also traced the historical journey, understanding that genes code for these chains.
Yeah, from Garrod's early insights through Beadle and Tatum's definitive experiments to Ingram's pinpointing of a single amino acid change, it all leads to this understanding of the four levels of protein structure, which ultimately create the incredible diversity of function we see in life.
An amazing journey from code to function.
Truly is.
Okay, we started by highlighting that huge discovery of the ribosome as a ribozyme, RNA, doing the catalytic work. So here's our final thought for you to ponder. What does it fundamentally mean that the core process of translating life's genetic code relies on RNA catalysis, not just protein enzymes? What might that tell us about the very origins of life on Earth and the capabilities of RNA itself? Something to think about. Until next time on The Deep Dive.